Hello, everyone. It's a great pleasure to meet you in virtual. I'd like to start the presentation with an acknowledgement to the conference organizers for them to provide a platform for us to share our research. Today, I want to share some of the studies that we have done to look at how engineered nanoparticles affect plant uptake of heavy metals and the underlying mechanisms for that. So many of you probably have seen this figure before. So this is a prediction of the global population, and we have heard that many alarming predictions that our global population will continue to grow, and some people use explosive growing or growth. So while we don't know exactly what will happen, but we surely know that the population will continue to grow, and we will need to feed them, provide them shelters to to live, and necessities to live. Because of that, the population is growing, and the arable land is dwindling. So the average land to support a person is becoming steadily decreasing. This requires us to come up with some novel technologies to increase our agricultural productivity. In the past century, the humans, we have come up with many interesting and novel technologies which help us to feed the world, feed the global population. For example, we have developed new technologies to fix nitrogen from the atmosphere. The machines are used more commonly in agriculture, which apparently increase the productivity. We have developed more accurate, precise irrigation systems. We have developed many agrochemicals which help us to have higher yield. So we have achieved quite a bit. However, when the time comes to about the beginning of this century, and what we have noticed is that, so the yield has been somehow plateaued, but at the same time, the environmental cost is becoming higher and higher. So this is not sustainable. So we must come up with new transformative technologies which help us to maintain the yield, but at the same time, have lower environmental footprint. So we have tried many things as a community. So we, for example, the biotechnology, transgenic or genetically modified crops, for example, information science, for example, new sensors and big data and so on and so forth. But now it appears that nanotechnology, which is an enabling technology, becomes one of the most proximate technologies to help us to achieve our goal, which is to maintain, at least, if not increase the yield, but at the same time have a lower environmental impact. So this slide shows you some of the potential applications for nanotechnology in agriculture. Nanofertilizers has been investigated. We're trying to develop slow-releasing nanofertilizers because the conventional solid-based fertilizers, if plants cannot use them, we could lose them with the runoff. Another uses are nanopesticides and targeted release of those materials or chemicals. Apparently, in order for them to release when we need them, you will have to provide some stimuli, and a pH, for example, temperature or moisture are common stimuli to tell the nanoparticle systems that now it's time for you to release the chemicals. That will apparently help us to save the pesticides or other chemicals, but also help us to maintain the environmental quality. So other uses including added nanoagrochemicals as soil amendments, or use that to fight crop diseases and so on and so forth. So the usage is very diverse, but one thing in common is that in many of these applications, it involves the application and the dispose of nanoparticles to the field. Okay, therefore, there's a big chance that we will be seeing more and more engineered nanoparticles in the environment, especially in agricultural settings. And the problem is that even before this nano technology, in which while it provides many promising applications, some people have a concern about the applications of nanotechnology will cause the accumulation of nanoparticles in crops and endanger human health. So those of the concerns has been studied extensively, but for this presentation though, we'll focus on another side of the concern, which is that before the nanotechnology field or era, there has been many of those chemicals in the soil, organic compounds such as fertilizers, pesticides, and heavy metals. So they are already a concern, and they can get into the plants and cause lots of issues. And many processes are involved in their uptake, and nanoparticles could affect many of these processes. For example, we use arsenic as an example because arsenic is a huge concern in some Southeast Asian or East Asian countries. So arsenic uptake is complicated. So arsenic also has a rich chemistry in the soil and plant rhizosphere, and many of those processes are involved in its uptake. So when nanoparts are introduced in, some of the metal are nanoparticles. They are also very active. Some of them are maybe photosensitive. If you have light, some of them could kick out their electrons, and that electrons could reduce certain redox sensitive chemicals such as arsenic 5 or arsenate. And for some of the metal oxide nanoparticles, in particular cerium oxide nanoparticles, and the copper oxide nanoparticles both have the strong potential for agricultural uses. On their surface, they naturally contains these redox couples, such as cerium 3 and cerium 4, or copper 1 and copper 2. So those redox couples could couple with arsenic to affect arsenic speciation, and which apparently will affect the metal uptake. And we have done years of studies to look at different nanoparticles and different metals, and we have found that nanoparticles often has a huge impact on the plant uptake of these heavy metal ions through different mechanisms. So in the following slides, what I'm going to do is that I will share some of our studies in the past, and then after that, we'll look into why nanoparticles could affect this plant metal uptake. So starting with cerium oxide nanoparticles and cadmium, because cadmium is a very common heavy metal, but it's very toxic, okay? So we started with a very simple study, which is the hydroponic study. We grow soybeans in liquid media. In the media, we add 1 milligram per liter of cadmium or 100 milligram per liter of cerium oxide nanoparticles, or both, okay? We have done this both in hydroponic systems and soil systems. I'll share the results from the soil system in a moment. So for the hydroponic system, after weeks of growth, what we have seen is that the joint exposure didn't really affect the biomass significantly. So there's no huge impact on the biomass growth. However, when we look at the concentrations of cadmium in plant tissues, so on this side, this cadmium concentration associated with the roots, when I say associated, it means that some of the cadmium could just absorb on the surface, others could be actually inside the roots. So this cadmium, we didn't make the distinction between those two fractions. So that's the total cadmium associated with the plant roots. And what we found is that cerium oxide didn't really affect the concentrations of cadmium associated with the roots. However, when we look at the cadmium in the shoots, here, the upground tissues, what we found is that the presence of serum oxide nanoparticles significantly reduced cadmium concentration in the shoots. So this could be because either because nanoparticles prevent the cadmium getting into the roots, or it could prevent or affect the transport process. So we'll look at that in a little bit. And then, as I said a moment ago, that we grew soybeans hydroponically, and we also grew soybeans in soil. And in the soil, we add either 0.25 or 1.0 milligram per kilogram of cadmium, which is fairly close or within the range of background cadmium in agricultural soils. And then we add 500 milligram per kilogram of serum oxide nanoparticles. And we let the plants grow for about a month. And we looked at a bunch of those physiological parameters. And here, what you are seeing here are the concentrations of serum and cadmium. Let's look at cadmium first. When we look at cadmium, what we have seen is that for both the roots and the shoots, we compare those two bars and these two bars, and these two bars, and these two bars. Nothing has been changed. So the presence of serum oxide nanoparticles didn't really affect the cadmium uptake in soil-grown soybeans. This is different from what we have seen in the hydroponic system. So we can look at the serum here. So if you look at the serum here, the cadmium also affects the serum, not in the roots, but actually, yes, significantly in the roots and also in the shoots for the ones which was 1.0 cadmium. So the 0.25 didn't affect the serum. So this is not the major focal point for this presentation. So I will not delve into the serum issue, but what you can learn is that the impact is mutual. Not only that serum oxide nanoparticles could affect the cadmium, the heavy metals could also affect the nanoparticle uptake reciprocally, okay? So while the overall cadmium concentration in plant tissues, the roots and the shoots are not significantly affected. But if we sort of look a little bit closer to look at the distribution of cadmium in plant tissues, we sort of divide the plant tissue into 4 fractions: the roots, relatively older leaves, and then newer leaves, and then the newest leaves. And then we look at the concentrations of cadmium and the serum. So on the top, that's the concentration of serum, and you can see that serum indeed is affected by cadmium, but our focus is on the cadmium. So this is the one which are exposed to this column shows the data which are exposed to, 1.0 milligram per kilogram of cadmium only in those different tissues. And this one shows the concentration of cadmium, which is exposed to both cadmium and serum oxide nanoparticles. Okay, if we look at the statistics, so there's not a significant difference. However, if we look closer to the trend, that does give us some idea that serum oxide nanoparticles could affect the distribution of cadmium. So when you have serum oxide nanoparticles, the concentration of cadmium in the roots is a little bit less. The first section here is also less than what you see, but then gradually you can see that there is a cross here. Basically, what that shows is that the presence of serum oxide nanoparticles somehow enhance the transport of cadmium from the roots to the shoots. And that could have the big implications for health if we allow this soybean to grow all the way to maturity. That could affect the concentration of cadmium in the edible tissues. So that's again an important observation, even though the absolute comparison gives you non-significant impact. And we also looked at how metal oxide nanoparticles affect plant arsenic uptake. So we looked at cerium oxide nanoparticles and zinc oxide nanoparticles. Different from what we have seen for cadmium, what we are seeing here in a hydroponic system is that cerium oxide nanoparticles had a very limited impact on the plant uptake of arsenic. Doesn't matter whether we introduced arsenic as an arsenic 3, the reduced arsenic, or the oxidized arsenic, arsenic 5. However, when we look at the zinc oxide nanoparticles at the same concentration, we are seeing a very dramatic significant decreases, except for this one here, the zinc oxide nanoparticles in the shoots, when the arsenic it was provided as an arsenic 3. It's still reduced, even though it's not significant. But for all other situations, zinc oxide nanoparticles significantly, reduced the plant uptake of arsenic. Okay. So for zinc oxide nanoparticles, for some metals they are more soluble, such as zinc oxide nanoparticles or silver oxide nanoparticles, silver nanoparticles. People were always debating whenever you see some impacts from this type of more soluble nanoparticles. The question is always is that because of the nanoparticles or because of the ions they dissolve? So to find out whether the dramatic reduction of arsenic by zinc oxide nanoparticles is because of the nanoparticles themselves or because of the ions release, we have done another set of studies. In this study, we also introduced arsenic as either arsenic 3 or arsenic 5, and then in combination or separately with the same concentration of zinc oxide nanoparticles and zinc ions. So what we found is that doesn't matter whether it's nanoparticle form or zinc ion form, they pretty much reduced the arsenic concentration in roots as what we have seen to the same extent. So like what we see here, there's not much difference. However, when we look at the arsenic in rice shoots, what we're seeing is that the reduction is a lot more dramatic for the zinc ions than the zinc oxide nanoparticles. So apparently, what that shows is that while both of them reduce the reduction of arsenic, the ion is just has a greater, bigger impact. So the nanoparticles. So with that said, the conclusion at this point is that the nanoparticles themselves could have some impacts, and that's different from their ions in terms of their impact on plant uptake of coexisting heavy metals. We also tried to see the nanoparticles versus ions in a more realistic environment, which is to grow them in actual paddy soils and the bigger mesocolumns rather than the smaller tubes. And the results are fairly similar, that for both the roots and the shoots, we're seeing that zinc ions is doing more impact, is having more impact than the zinc oxide nanoparticles. And we have also done arsenic and examined how copper oxide nanoparticles, because this again, is a fairly common nanoparticle with great potential for agricultural use. And the results are fairly similar to what we have seen for zinc oxide nanoparticles, which is that overall the nanoparticles reduce the plant uptake. This case is the rise uptake of arsenic, regardless of the initial oxidation state of arsenic, but the copper ions in general shows greater, stronger, more impact on the arsenic impact uptake by plants, except for this one here, when your initial arsenic is arsenic 5. So we have done many more. So we have looked at other types of metals, such as lead, used other plant species. But the conclusion is very consistent that when you have engineered nanoparticles in the plant heavy metal system, those engineered nanoparticles could have dramatic, significant impact on the plant uptake of coexisting heavy metals. Okay, so this is the number one, or the major lesson we have learned. So we have observed through this many studies, but the more important question is that how and why? To answer that question, I will use cadmium and arsenic as an example to show you how engineered nanoparticles could affect the plant uptake of these heavy metals. Okay, so first of all, let's start with the plant uptake and accumulation for cadmium. So cadmium in the environment, how did they go through the many processes to get into plants? So this is not a simple one-step process. It involves many, many different intermediate processes. Okay. So first of all, the heavy metals in the environment, in many of them are adsorbed on the soil particles. They will have to desorb from particles to get into the pore water to become available. So after getting into the water, the first thing they do is that they need to attach or adsorb on the root surface. Okay, and then they will find a way to get into the plant tissues. And in general, there are two pathways are involved. One is called the apoplastic pathway. Another one is called the symplastic pathway. So what that means is that for the apoplastic pathway, these particles would go with the water flow and just pass through the cell walls, because that's the place where you have the least resistance to get into plant tissues. And until they get into here, so this is called the endodermal layer. And as you can see that between two cells, there's a impermeable Caspian strip that pretty much seals the plant. If anything wants to get into here, the diamond tissues, so that's where the water will be further transported up to the shoots. If you don't get into here, you will be controlled risk to the root tissues, okay? So in order for you to pass that, you will have to pass the membranes, otherwise it's sealed. So the second approach, symplastic pathway, is that the water and whatever the contaminants will go through the layers of the cells, and in each cell layer, they will have to pass through the cell. And when they pass through the membrane for metals, the root membrane has many, many transporters, which actually controls who's in and who's out, okay? So those are the processes. Now in here are the heavy metals, right? Those heavy metals, those plants for normal physiological function. So there are many microorganisms. Those microorganisms are supported by the carbon source from the root exudates. So those root exudates could form complexes with these heavy metals. And that apparently, if you look at the transporters, it is one thing that these transporters, there are many specific transporters. Some of them are just for the chelates and others are for freestanding cadmium. Therefore, if the chelation process is affected, that could affect how many of that will be getting into the plant tissues, okay? So all of these processes together determine how much cadmium is getting in, And when you introduce engineered nanoparticles to those process, to the system, engineered nanoparticles could potentially affect all of these processes. And in fact, our research has shown that engineered nanoparticles do affect these processes. Starting from the first one, what we have found is that engineered nanoparticles could affect cadmium bioavailability. Okay, so I will use zinc oxide and cerium oxide nanoparticles as an example. We have done quite a few nanoparticles. So what we have seen is that for zinc oxide nanoparticles, so we add with low concentrations, they somehow inhibit cadmium uptake. But when you have relatively higher concentrations of zinc nanoparticles or ions, they actually enhance the cadmium uptake. So what happened? So what happened is that when you have a relatively lower concentration of cadmium, zinc and cadmium, if you look at where they are in the periodic table, they are in the same column. Their properties are fairly similar. So the zinc and cadmium are actually competing through those channels, who's getting in. Therefore, that somehow reduces the cadmium uptake. Okay, but when you add a lot of zinc oxide nanoparticle ions, so the zinc oxide nanoparticle releases ions, and these ions, you need to find a place for that. They will adsorb on the surface of the soil particles. They actually competing with the cadmium and the adsorption sites, and they actually can replace some of them, switch some of the cadmium, so make them bioavailable. Therefore, when you add more of the zinc oxide nanoparticles, we found that they actually increase the cadmium bioavailability. Okay, so but there are also other mechanisms depends on the particles. For example, cerium oxide nanoparticles could potentially reduce or lower the bioavailability. A reason is because that this nanoparticles has a very large specific surface area that cadmium could adsorb on the surface, and our previous research have shown that cadmium cerium oxide nanoparticles showing fairly good capacity for cadmium adsorption, and apparently that will be affected by the root exudates, and because root exudates could form complexes, and the adsorption capacity for freestanding cadmium and the capacity do differ. That's the details, but the overall concept I want to convey through this slide is that nanoparticles could affect the bioavailability of heavy metals through different mechanisms. And the nanoparticles could affect the plant's apoplastic pathway. So you recall that for the apoplastic pathway, the heavy metals is getting into the plant tissues with the water flow following the cell wall paths, right? So they will not be blocked until they hit the endodermal layer. However, when plants are stressed, when the plants sense that there are probably some hazardous chemicals, metals, or nanoparticles all together, if plants sense that, they will start protecting themselves. And the one way for them to do that is to form this exodermal layer of strips. So they could seal the space in between the cells, and this could also deposit a layer of suberance around the cells. See the yellow color? So that's a layer of suberance. So suberance are much less porous, much less permeable, so that potentially blocks the entrance of heavy metals into the plant tissues. We have done some of those. We take the root samples after they are exposed to different treatments, heavy metals alone or the combination of metal plus nanoparticles. And we have used a fluorescent microscope to observe how tightly they're sealed from where they're starting, and so on and so forth. And what we have found is that, indeed, when you have the co-exposure, when you have serum oxide nanoparticles together with cadmium, it affects where the calcium strip is built and whether it's fully closed or only partially closed, and so on and so forth. So if, for example, if we can compare that, say Cd, the cadmium 0.25 milligram per kilogram of cadmium alone or with serum oxide nanoparticles, apparently these are significantly different. The same thing is true for the cadmium at 1.0. So basically, what happens is that when you have nanoparticles, some of serum oxide nanoparticles are protective. So they could potentially scavenge some radicals. Therefore, serum oxide can provide a certain protective role. So what we are thinking here is that when you expose cadmium with serum oxide nanoparticles, so the serum oxide somehow scavenges part of those radicals in the plant tissues caused by cadmium or some other things. Therefore, plants feel more relieved that they don't have to build a deep barrier. So apparently the benefit for plants is that now when you have large spaces, they could have more space to take up water, take up nutrients. So that's better for the plants. So we can further look at the meaning of why this is relatively shorter. But the thing is that when you have less of that, so the water uptake could be much higher together with the cadmium uptake. So the whole point is that the nanoparticles could affect the plant root anatomy and therefore potentially affect cadmium uptake. In addition, we have found that nanoparticles could affect the plant physiology. So if you look at the plant cells do have these pores, which allows the transport of the materials between cells. When you have both serum oxide and cadmium, the pore sizes are different. So it looks like the pores, because of the serum oxide nanoparticles, maybe some synergetic effect. The pores are a bit tighter. So that apparently will affect the flow of cadmium together with water. In addition to that, what we have found is some nanoparticles could just simply because of toxicity, which limits the plant uptake of water. Therefore, together, they affect the apoplastic pathway of cadmium uptake. So nanoparticles could affect cadmium speciation in plant roots, because that what we have seen is that when we have this joint exposure of serum and cadmium to soybeans, that somehow encourages the release of root exudates, in which we have shown by higher concentrations of total organic carbon. And because many of the root exudates are amino acids or small, low weight, molecular weight acids, therefore, higher release often means lower pH, and that one has also been confirmed that when you have co-exposure, that changes the chemical environment by facilitating or by stimulating the release of root exudates. And that has a big impact on cadmium uptake, because the cadmium is a freestanding cadmium. Cadmium could form some complexes with hydroxide ions, and that formation, so this is not soluble, okay? Could potentially be persistent as particles. Therefore, the relative distribution of those different species does affect the availability of cadmium. And for this, the pH is a major factor here. So in addition to that, those root excites are all molecules, organic molecules, and they do have this nucleophile groups or more electron rich groups. They could form complexes with cadmium ions. And when you form complexes, it's just one thing we know is that once cadmium is complexes, they will have to go through different channels to get into the root cells. And apparently, the efficiency, the control of those channels differ for free cadmium and the complexes. In addition, for some of the engineered nanoparticles, they could induce reactive oxygen species, and they could also, this reactive oxygen species could affect the expression of these proteins. Therefore, they affect the simplest pathway of cadmium uptake. So now I want to share a little bit of results about arsenic uptake and how engineered nanoparticles could affect arsenic uptake, because arsenic, cadmium has relatively simple chemistry. Arsenic has more rich chemistry. Even though many of the processes are the same, one thing which differs for arsenic and cadmium is that the arsenic has a experience lots of chemistry in plant roots, root areas, and that has a huge impact on how much arsenic will be able to get into the plants. So what we have found is that when we introduce nanoparticles to the plant systems with arsenic, the plants, the engineered nanoparticles could affect the arsenic speciation. And this is one of the results we have obtained, is that when you add either zinc oxide nanoparticles or ions into soils with arsenic, first of all, in the case, doesn't matter whether you have arsenic or not, or no, sorry, whether you have a zinc oxide nanoparticles or not. Arsenic 5 is the dominant species in all treatments. However, when you add zinc oxide nanoparticles or ions, what we are seeing is we are seeing a good increase of the arsenic 3, because the arsenic 5 to arsenic ratio have been significantly decreased from 55.6 in the control group to 17.9 and 23.8. That's more than 50% reduction of the ratios. And then we also found that the organic arsenic, even though that's not a minor, that's not a major species in many occasions, but in this case, the introduction of zinc oxide nanoparticles significantly increased this DMA, dimethylene arsenic, and while the zinc ions appears to be more beneficial for the monomethyl arsenic, the MMA. So that clearly the arsenic speciation has been affected by zinc oxide nanoparticles or zinc ions. And therefore, as we have seen that the arsenic speciation plays a huge role in how much of the metal can get in, therefore, the nanoparticles could affect arsenic by changing the speciation of those metals in soil. That apparently will lead to the arsenic speciation differences in plant tissues. And that's exactly what we have seen. For example, here, so the top ones are the ones when we introduced arsenic as arsenic 3. And what you can see is that I will use serum oxide nanoparticle as an example here. So this is what you see in the rice roots. And what you are seeing is that the co-presence of serum oxide nanoparticles didn't really affect the root concentration or the speciation of arsenic in the roots with the controls. However, that does affect the speciation in the shoots. Okay, and for others, when you have arsenic 5, what we can see is that the serum oxide nanoparticles increased concentrations of arsenic 3, but decreased the concentration of arsenic 5 in the roots. So we can look at further of the speciation, but the lesson here is that the nanoparticles, they affect the speciation of arsenic in the root rhizosphere, and they also affect the arsenic speciation or distribution or transport in plant tissues. If you look at this one here, for example, the arsenic 5 did not significantly differ from the arsenic 5 in the treatment with only arsenic 3. However, the arsenic 5 here, actually, it's kind of close. That's a bad example. But somewhere here, so we can look at the zinc oxide here. So you can see that arsenic 3 is dramatically different, but here arsenic 3 is less dramatic in the shoots. That means that zinc oxide nanoparticles could potentially affect the arsenic 3 transport. And the mechanisms behind that is very complicated, and that's something we are still trying to figure out. But the lesson is that the nanoparticles affect arsenic speciation. So this is a slide you have seen before that arsenic affect the zinc oxide nanoparticles or zinc ions affect arsenic uptake and transport. So here we can, I did a little calculation here to calculate the total arsenic in root and shoot ratio and calculate that ratio. Clearly, what we can see is that for the nanoparticle ions, okay, the impact of zinc oxide nanoparticles on arsenic is not limited to the rhizosphere and roots, but it actually affects the transport of arsenic inside the plants. So if you look at the ratios here, the zinc oxide nanoparticle, because of that, the transport is twice of what you see in the arsenic treatment or the one with the zinc ions. So basically, what we are seeing is that both zinc oxide nanoparticle ions reduce arsenic uptake in the rhizome roots, as what we have seen before. However, the nanoparticles somehow benefit the transport. So that's why we have seen that they have the similar impacts on the roots, but the impact on the zinc oxide was deeper. Actually, it's not because the zinc ion has a deeper, greater impact. It didn't really affect the transport. It's because the zinc oxide nanoparticles then helped the transport of the uptake of arsenic. So again, from this many studies and ions, we can see that zinc oxide nanoparticles and other metallic nanoparticles affect plant uptake and the transport of heavy metals. Okay, so before I conclude the presentation, I want to share a little bit ideas about the nanoparticles versus their ions, because these are the issues a lot of people always debate and ask. So for zinc oxide nanoparticles, it has a very high solubility. The dissolution rate is very high. It will give it time, maybe hours, or maybe a day. All of them could be just zinc ions, okay? Then if that's the case, then really the impact is how nanoparticles reduces ions and then have some impacts. if that's the case, then why we're still seeing the differences of nanoparticles versus ions? I think one of the reasons is the kinetics. Okay, so while the zinc nanoparticles could release ions, and those ions could also form other complexes with, for example, hydrogen sulfide or phosphate to form other types of nanoparticles. But in the ion case, ion is all of the ions are there. So this connection will be much faster. But for this one, it's a little bit slower, so that in the beginning, this is all you see, but in the nanoparticles, in addition to those different transformed products on ions, you also see some nanoparticles. So the cocktail have different components, even though eventually they will become the same. Therefore, that's why that even for those highly soluble metals, you would potentially should expect some potentially different impacts with their ions. And for that, the in situ characterization of the zinc ions or other soluble nanoparticles would be very important. So with that said, I can conclude my presentation that the key information, take-home information is that engineered nanoparticles can and do alter the plant uptake of heavy metals. And this could be done through many different processes. The nanoparticles could affect the plant, affects the bioavailability of heavy metals to plants through different processes. One, their competitive adsorption to soil particles, and the adsorption of those heavy metals on the nanoparticles themselves, or the modification of the metal speciation in plant root zone. All of these three processes could contribute to the changed different bioavailability. The nanoparticles could affect the apoplastic pathway of heavy metal transport by either affecting the root anatomy or the root physiology, including the membrane pore sizes, or maybe just the toxicity impact, which literally reduces the plant needs of the water uptake. And the nanoparticles could also affect the symplastic pathway because they could affect the speciation of the cadmium arsenic, and that will depend on which channels they used to get in, but they can also potentially affect the expression of heavy metal transporters. Okay, therefore, these are all processes. Some of them we know why, others we are still trying to figure out how these processes are affected by different nanoparticles and different heavy metals, their combination, and how we can associate that with the nanoparticle properties. So many questions remain, but at least we have identified several major processes that ENPs could play a role in plant uptake and accumulation. So with that said, thank you for your attention, and I hope you all enjoyed the presentation. And I will close the presentation with my acknowledgements to all my lab members, previous students or current students, my collaborators, the funding agencies which helped to make this research possible. And again, thank you all for the conference organizer and all the audiences for the opportunity to share my research with you. I'll be happy to answer any questions. If you have any, feel free to send me an email. With that said, I will close the presentation. Thank you. Bye-bye.